Hello, everyone. So before I get started, I'd just like to say that I'm representing Collaborative Drug Discovery, or CDD for short. For those of you who are here at the conference, you've probably seen my colleagues in the exhibition hall. If you see anything interesting, don't be shy. Do feel free to... Oh, someone scored a goal. Okay. Okay, so by way of introduction, in the world of drug discovery, we have huge amounts of data that exists really on both sides of the quality spectrum, shall we say. So we have whole disciplines like cheminformatics and bioinformatics, which are very mature. They have a long tradition of doing great jobs of representing molecular entities in a way that's both machine readable and human readable. So there's a lot of good stuff there. And so what we find is very often is that we have two or more data sets that are consisting of, say, collections of small molecules that are very well represented. We'll have an activity measurement of some sort, which is just a number and units and some other metadata. And then we have the description of the actual experiment that was used to create this data, what they measured, how they measured it. And I've drawn them on screen here as post-it notes because really for all practical purposes, they might as well have just scribbled it down on pieces of paper, because the only way to really be sure that two assays are comparable is to have an expert read them and then pronounce a decision as to whether they're the same thing. Now, obviously, that really doesn't scale well at all, and it causes all kinds of other problems. If you peer under the hood a little bit and see what people actually do out there, for the most part, within organizations, they tend to very often have their own bespoke electronic lab notebook system where they'll perhaps we have one or two fields that are linked to an open public database, which is the ideal situation, but then the rest of the content will be the usual mishmash of free text, custom pick fields, file attachments. And even if an institution is using these in a very rigorous way, they're almost never doing it the same way as any other institutions. So the interoperability of this data is really very, very low. And this is a huge problem. As you may not be surprised to hear, this is a natural match for using ontologies to solve some of these serious real-world problems. Now, if you get started at trying to use this approach, well, first of all, you'll find that nothing is ever simple. And so you need a number of ontologies to describe these assays thoroughly. So some of them are very broad and designed for this purpose, like the bioassay ontology. And then you have others like cell line ontology and protein ontology and so on that go very specifically. They get huge, vast, enormous trees of data. And then you have others that just contribute a few terms here or there, and you've got overlaps and so on. So the point is, if you're actually describing bioassay protocols, you've got some tools, annotation tools that are not very easy to use. You have to have a very high level of understanding, and it's very hard to do anything consistently. So I like to say it's a little bit like handing someone a dictionary and asking them to write a novel. I mean, the point is, the results are not really going to be very good. So we took a pragmatic approach to this. We embarked on designing a template outline, which ended up being a couple of dozen fields that capture most of the information that you need to know about bioassay screening experiments. And for each of these fields, it basically represents a subsection of the relevant ontology. So it sort of organizes everything into a reasonable form and makes it a little bit more manageable for people to actually do the same things in a consistent way. Now, just show a couple of screenshots of what this template editor looks like in real life. This is a tool that's used by people who do know a bit about semantic web. Almost everything else I'm talking about is designed for people who are not really that interested in the subject. They're just going to use it. So the first thing to do is to organize these series of categories, and each of these template categories is linked to an IRI under the hood, so that the categories themselves are already part of the semantic web. And then for each one of these, the key is to select which part of the overall collection of ontologies applies to that category. So sometimes it's pretty straightforward. You just have to find the best root branch and say, this is where your content is coming from. But other times you have to pick and choose between multiple parts of multiple templates, do a bit of pruning and grafting and so on. And our template model handles all of that. It can be a little bit fiddly sometimes. But the important part is that once this actually gets compiled and presented to the end user, it just looks like a very ordinary hierarchy user interface. It just has a series of labels that should all be familiar. On this right-hand side panel, I mean, even from the back, you can probably see that there aren't any IRIs on the main screen. You don't see any semantic web stuff. It just looks, I wonder who scored that one. It just looks like a very ordinary selection panel. So you only get this semantic web information when you mouse over something. It sort of gives the game away. But there's a recurring theme here is that the end users don't need to know that they're using this fancy new technology. They just benefit from it. Okay, so at this point in the story, we've basically got a collection of ontologies, a template, a tool for annotating them, but this is a long journey and there's a lot more to do. So that the next thing we set out to achieve is to create a significant body of content so people would have examples of how to annotate at real-world assays from public data using this tool. And this also corresponds to a grant-funded project we had to develop machine learning models to accelerate the curation of an actual human, not replace a human, just speed them up. Should note there, this is around 10 years ago, so this precedes large language models. So the models were good for their time, and we were able to, our experts were able, domain experts were able to power through thousands of assays, which were done in quite a lot of detail in a surprisingly short amount of time. And actually all of this information is now available online. You can use it to do whatever you like for your own nefarious purposes. Now, all of this exploratory research and development was done under a project called Bioassay Express, and this is now open source. There's a web online version that you can play with, but you can also go to the GitHub page. There's one Google search away from it. And this project, it does a lot of stuff. We really took a lot of ideas and pushed them really far to see what we could do with them. And I just want to zoom in on one in particular that gives you an idea of why ontologies are actually so much more useful than any other strategy. So the feature of choice here is called the explore page, and it's basically you add a series of constraints, so you just filter down your list of assays until you've got the list that you're interested in. So you may not have a clear idea of what you're looking for. The first constraint that I've added here is basically to say that I only want to look at binding assays. Now, this seems like a very simple thing to do, and it is, but what we're doing here is the query itself, it has all of the context with it. So the right category, the right value, both based on IRIs from semantic web, and the data itself is annotated according to that same scheme. So that basically means your query is both precise and accurate. So there are no false positives, no false negatives. I mean, there could be errors in the curation, but other than that, you get exactly what you asked for, nothing more, nothing less. This is in contrast to, say, a keyword search, in which case you get all kinds of stuff you don't want, and you probably miss some stuff that you do, or even a more sophisticated language analysis approach. You still wouldn't get everything, and you'd lose some good stuff. So some simpler approaches may be good enough for interactive, but we're really talking about designing something that could be used by machines as well as humans. Okay, so continuing on with this example, I've added another set of constraints. So I want to say that the organism of interest has to be a rat or a mouse. And again, just like I was saying before, this has context with it. It's not just a document that's talking about rats or mice. It's saying the organism of interest on which the biology applies has to be one of these two organisms. So again, you get a very precise query. Okay, now this has now narrowed down the list of assays to something much smaller, and you probably are wondering what that is. It looks like an arcade game from the 1980s, but if I zoom in, I can explain that this is a way to digest a large quantity of information in a relatively small area. So in the columns there, we have the assays that match the query, and on the left, we have in the rows, we have the individual categories from the template. So what I'm showing here is the assay format block, and you can see the rows are organized in a hierarchical form. So these are the terms that apply to the selected assays. Now, what you can hopefully see in there is the hierarchy, and this is something that we get for free from these ontologies that other people have built for us. And so what I want to emphasize is that the square blocks there, that means the curator has asserted that particular ontology term applies to that assay. And the narrow bars there, they show the inheritance hierarchy. So we can see that there's two annotations that share the same branch have more in common than those belong to different branches. So we get a lot more information than just this thing was asserted. And also for those of you who are informatics minded, you may be looking at this grid and thinking that you could represent either the rows or columns as a vector of zeros and ones and construct a kind of similarity metric. So you could come up with a quantitative metric for how similar two assays are to each other. And we have done a certain amount of work with that, so we can come up with a number that'll tell you, you can rank assays according to similarity with that. So that should hopefully give you an idea of how we can get some pretty powerful search functionality once we've actually got a body of data that's using this content. All right, so now I want to come back to a bit more of the big picture. And so the system for creating content is fairly dysfunctional. We tend to start on the left here and go through the top pathway. So at the very beginning, we have a scientist who knows as much as will ever be known about how the experiment was constructed and what observations were made and the results that come out of the instruments. And then typically that scientist will be tasked with the job of downgrading that information into something like some kind of text and figures combination. And then that will be placed in a database of some kind. And then if we want to do informatics with it, someone has to come along and re-upgrade that data to some more machine-readable form. And that's, at this point, we have two really not very good choices. We can lean in heavily to machine learning and accept that we're going to create a lot of extra mistakes that really didn't need to be there. Or we can pay experts to recreate the data well, but that's slow and expensive. Now, of course, this is kind of a straw man argument, but wouldn't it be great if scientists were to just annotate their data directly into this highly machine-readable format, which is also very highly presentable to humans as well. There's no problem there. But the key issues, of course, we need very specialized, well-designed software for that, which we can do, that's no problem. But the harder part is that people may need to make, or scientists specifically need to alter their workflow. And if anyone has ever tried to do that, you will know how brutally difficult that is. But nonetheless, at CDD, we are up for the challenge. Okay, so now the second phase of this whole long-term project is that having built this sophisticated open source prototype, we're now turned our attention to how to integrate some of this functionality into our software as a service product, which is called CDD Vault. And we've really taken an 80-20 approach here. So we learned a great deal about which features were really interesting to everyone and which ones were kind of nice to haves. And so we're trying to help people get into this ontology annotation without disrupting their workflow at all, if possible. And so we're trying to be a little bit stealthy and subversive about it whenever we can. And so we've introduced another layer, which we call protocol forms. And this allows individual users or group leaders to create subsets of all of the data that people normally use to describe their protocol. So we have thousands of scientists who are doing this every day, so that's an advantage that we have. And they're using the traditional approaches, text, drop-down list, that sort of thing. And we can allow these templates to just incorporate one or two ontology sections. So you can imagine perhaps before you had a field called organism, maybe it was a free text value, just type in rat, dog, cat, whatever it is. Or maybe there's a drop-down list, you have to add a new animal as text whenever it comes up. Now you just pick it from the ontology section and all of those organisms are just there in a big hierarchy. And not only is it done for you, but these are standardized across the entire universe of drug discovery in principle anyway. And it's very easy to pick the right one that you want. Everything works. It's kind of like what you had before, only better. That's what we're going for. And when it comes to actually filling these things out, editing a template and filling it in, it's very much the same thing. So in this case, filling out with an ontology is very much just like a drop-down list. Now, thinking more about these forms, the ideas that we're going with, and this is mostly future work that we're going to be doing soon, is that you can have as many forms as you like. They're easy to make. And a lot of work in science is quite repetitive. So if you find yourself doing very similar assays over and over again, it's quite likely that you'll want some default values because you just get sick of filling them out all the time. And if you've got several different types of projects across a group, you just have lots of different forms. We can be a little bit more strict for that, thinking more like imposing actual requirements. So we could lock those values in. So for example, we could say, this form is the one that you use when the organism is a mouse. If you're working on a different organism, you should be using a different form. So we could lock in certain values, and that's just how we do business here in this particular group, say. Or another idea is to restrict certain protocol ontology areas to a sub-branch. Some of these protocols, so some of these ontology branches are huge, like every protein, every disease, etc. It's kind of annoying to go through them all every time. So for example, this form was just for variations on pancreatic cancer. We could just say, only show that branch. So people, they can't select from anything else. And what they want is easy to find. So you get this dual effect of, it's harder to make mistakes, but it's also easier, it's quicker. So, and there are some other things that we can entertain, because a lot of this data is kind of codependent. And so for example, if you select an instrument type, that instrument is not going to have every possible type of detection method, probably just going to be limited to one or maybe several. And so that second category can be limited based on your choice of the first one. So where we're really going with this is we're really trying to make it so that creating this data is much easier, but also less error prone. So we're really trying to encourage people to create this very high quality data with actually less work, not more. So the pitch is really not that you have to wait for the network effects to kick in after you've done all of this extra hard work. Actually, no, it's easier. And then the network effects will kick in and make everything really cool. So just saying to wind down, really all of this, where we're going with this ontology annotation, it mostly comes down to collaboration, so at multiple different scales. And so on one end of it, we have a lot of people talking about contract research organizations. That's a hot topic right now. So if you're able to communicate with people you've never met and tell them exactly what you want, and they can communicate back and tell you exactly what they did using the same vocabularies, then there's some pretty obvious advantages to that. But then again, at a smaller scale, there's the preservation of institutional knowledge. Like if somebody moves on to a different institution and their experiments are still sitting around in a database, they're no longer dead data. If it's annotated well, according to vocabularies that you used yourself. It's also very easy to see when you've missed something. You just do a simple report, and if you see a whole lot of blank areas on the grid, then you need to come back and fill those out. And one thing that I also like to think back to the days when I was a scientist is that you're also kind of collaborating with your future self, because you know that you're gonna forget certain things, and you remember what you were doing exactly 2 years ago. Who knows? So if you can fill them out with a very usable information, you're actually helping your own research as well. Okay, so last content slide. In terms of where we're going with all of this stuff, it's we'll be in this for a while longer. In terms of curation and searching content, we're pretty happy with the way things are working. That slide I just showed about business logic and forms, that's coming up soon. One of the things that the actual template editor, we haven't exposed that even to power users just yet, because it is possible to make a mess. I mean, that'd be kind of annoying. So we're going to train people up to use that before we invite people to create their own templates. Another thorn in our side is that it's quite difficult to modify the underlying ontologies retroactively. And I would invite you to perhaps get in contact with Hyundai, who worked with me when we were looking into this problem. It's a difficult social and technical engineering problem, and it would be great to make some more progress on that. And another thing that we'd like to come back to is we got a lot of feature requests very early on for starting with semantic annotations and converting that into text descriptions. And it turns out that's a lot easier than going the other way. People really don't like writing up text, which is pretty understandable. There's another subject that comes up a lot and interacting with journals, so encouraging people to submit annotations along with their papers, but that's really a long-term thing, the issues of who's going to pay for it and how do you force authors to do more work than they do already. But we'll get there eventually, and it will be a great benefit when we do. And lastly, we have some grandiose ideas about how we can really use these extra levels of data about how similar assays are, that will be able to make the actual precision for structure activity modeling and drug discovery really quite a lot more effective. So finally, I'd like to thank the Vault team, the Vault and Bioassay Express teams. Pandey is one of the latter in the past time, and thank you for listening. I'd be happy to take questions.
So if you have a question, please come up to the microphone.
I was wondering, when you were surveying the landscape of available ontologies to design the model that you're using, what proportion of missing vocabularies or ontologies did you actually find would remain to be created or extended in order to express everything you want to get?
Yeah, that's a good question. For our common assay template with a couple of dozen fields, we were able to find ontologies for most of the things that we needed. So to cover most of the ideas, we actually mainly started with the bioassay ontology, which probably covers about 60%. Because it was designed for that purpose, but then we had to look further afield for specific cell lines, diseases, that sort of thing. And during the curation phase, we found it was the ontologies were already several years out of date by then. So there were new kinds of instruments, new assay kits, new this, new that, the other thing, and we did create a number of temporary terms in order to annotate those, but. For the baseline concepts for common high throughput screening, the coverage of concepts was pretty good. It wasn't too hard to find most of what we needed. We did dabble in some much more sophisticated ontologies, which had a lot of information about what materials you used, what order you did them in, and then we found a lot more gaps. We were trying to make more detailed versions.
Okay, thanks.
Thanks for the talk. I was just wondering to what extent it might be useful to try to read things basically directly off whatever the instrument config is, and how much you could actually fill by default from sort of the setup versus really requiring people to fill in as part of their experimental documentation.
Right, I think we did start working on that. We got distracted, but the idea was to analyze primarily the inputs for the robotic controls so that they could be mapped directly into ontology terms. So I think there's wonderful work that is yet to be done there.
Okay, cool. And as sort of a follow-up to that, do you think there's sort of an unfortunate incentive to keep things very proprietary and siloed across that might interfere with sort of ontologies like this?
You mean with the robotics or the ontologies?
With the robotics, but also just like people who make assays.
Right. I'd say with the robotics, probably yes, we did have some difficulty. In the ontology world, nobody seems to have any interest in keeping anything secret. It's sort of it's open by design from the beginning to the end. So that part has been really good. The difficulty is getting people to continue to maintain them, but that's another story.